So welcome everyone to our webinar on oncology. We are going to focus specifically today on clinical supply chain, of course, and how, well, we believe that clinical supply chain in oncology is actually a very, very complex task to succeed on. The objective of the webinar today will be to go through all of these sources of complexity, as well as some solutions that we want to share with everyone in the room today. Before we get started, I'm going to give you a couple of indications on how to use the platform which we are on today, Demio. On the top right of your screen, you will see a little arrow that is actually used to open and close the chat box. So if you click on it, you will have the chat popping up on the right of your screen. That chat will be used throughout the session. First of all, you may ask any questions that come to your mind in the chat. We will be answering them at the end of the session. For privacy, the chat is private, so no one can actually see what you will be writing in the chat. So feel free to really be completely candid in your questions as well. You also see a tab called polls in that little window. Whenever we have a poll that we want to share with you, it will pop up on your screen for you to answer, and the answers will be visible in that tab. After answering the poll, you can come back into the chat to keep on asking us questions throughout the webinar. If you're experiencing any technical problems in the webinar today, you can reach out in the chat as well to our technical facilitator, Miguel, and she will be able to help you with connection issues, sound issues, or whatever. Now let's meet the speakers. Let me first let Marine introduce herself.
Good morning and good afternoon, everyone. Thank you, Amaury. So my name is Marine. I am the regional manager working at Inside, responsible to develop our activities and potential partnerships in the APAC region. I'm currently located in Singapore, and I'm looking forward to this session with all of you. Thank you.
And I myself am Amory. I'm here specifically to share my experience in the past 8 years that I've actually been working at Inside specifically on oncology trials. I'm very happy to be here and share a bit of that experience with you. So what will the agenda look like for this webinar? We're going to start by discussing why oncology trials are so complex to manage in clinical supply chain, before having a quick reminder on why we're doing this and why it's so important to have research in oncology. We will then follow up with some solutions to manage that complexity and make our oncology trials more successful, and illustrate all of those solutions with case studies that we've worked on in the past years. And at the end, we will summarize all of the key takeaways and lessons learned that we share with all of you, and have a Q&A session at the very end. So let's start with complexity. What makes oncology trials complex to manage? I think it maybe as a first step, let's first agree all of us together. Is it actually that complex? I just want to see if everyone in the room agrees with that statement, that oncology trial supply chain is a complex thing to manage. So I will make a poll appear on your screen now. You should be able to see it. Do you agree or do you disagree with this statement? Okay, so 100% of answers on I agree. Well, I mean, somehow it's good news because it means that this webinar is going to be useful. Thank you very much for your participation. I see almost everyone has answered, so we can move on to the next question. On our side, at Inside, we also observe that oncology is indeed a complex area. When we start working with a company, we generally propose to perform a waste audit. So we analyze all of the clinical trials within that company's portfolio and see what is the amount of drug waste that we have, depending on the indications, therapeutic areas. And one interesting highlight is that oncology is actually one of the most wasteful indications in terms of IMP in the clinical trials world. So it does somehow highlight that there is a lot of complexity in the area. But why? Because the supply chain in the end is pretty similar. All clinical trials in the world, except maybe for cell and gene, have a very similar supply chain. We have to manufacture a drug substance or product, package and label the drug, distribute it to depots and then to sites where they're consumed by patients before being reconciled and destructed. So why is it so complex if the supply chain is very similar? Well, a first indication is it's very likely most of the complexity actually originates from factors that are outside of the supply chain's control. So let's maybe have a second participation from all of you in the room. Let's use the chat now. Can you write in the chat what you believe makes an oncology trial so complex to manage? I'd love to hear from you and what's your point of view on that question. So we're going to leave a couple of seconds for everyone to start writing down some answers in the chat. See some answers are starting to appear. So we see, for example, the uncertain dropout rate of patients. That's a very good point. A lot of investigators' choice, event-driven treatment duration, absolutely. Investigators' choice is a very popular answer, indeed. I see also multiple treatments. It's true that oncology trials have a lot of different treatment types. There's also multiple doses. Comparator sourcing can be a very complex endeavor due to, well, I know there's some shortages happening now and then. There's definitely a lot of things. I mean, we had already compiled in a slide a lot of ideas of things that make oncology trials quite complex to manage in the supply chain. And we like to actually organize a bit our thoughts. So we're using our very classic graph of what makes a trial complex, our drivers of complexity, to really talk about the specificities that make oncology trials complex. So if we're looking, for example, at the trial design, some of the ideas that were mentioned in the chat are very true. We have trial designs that are a lot more complex than other types of trials. For example, the length of treatment is completely uncertain. Patients could stay for years on treatment when for other trials, we have a very clear date for the end of the treatment for every patient. We have multiple treatments, generally standard of cares, chemotherapies. We might have actives and placebos, of course, and safety medications. We might have multiple dose levels, weight-based dispensing, body surface area dispensing. There's a lot of things that make it complex in trial designs in oncology, which means we're going to have a lot more unexpected events as well, because due to this trial design, we have now a possibility for patients to titrate more or less, to be heavier, to be surviving longer. On top of recruitments that can be faster or slower, of course, we have additional events that can be completely unexpected. And that makes clinical supply chain planning a lot more complex as well, because we have to make a planning based on uncertainty, which is very hard to do. And we have to do it on a much higher number of treatments. There's not only active and placebo generally in oncology, we have a lot more treatments than that, except in monotherapies. If we're talking about IRT, it also means we're going to have to use a lot more features of the IRT system just to have a safe setup and monitor a lot more features as well. And if we're talking about the supply chain, oncology is generally also using biologics, which we know have shorter shelf lives, are more complex to manufacture, have lower yields in manufacturing. So we might actually have more chances of having stockouts. So yeah, a lot of things really increase the complexity in oncology trials these days. Some of the highlights, just to quickly summarize what we said, we know there's a higher patient treatment uncertainty. We have more complex designs that can also be multi-indication. If you're looking at phase ones, or generally basket trials with multiple cohorts that are multi-phase also sometimes, phase 1 and 2 being merged into the same protocol. There's also a lot more adaptive trials in oncology than other indications today. We also have to look at comparator sourcing with extremely high costs generally, or even face shortages of such comparators and chemotherapies. If you go on the FDA's list of drugs in shortage, you will see that today 16 drugs that are really frequently used as chemotherapies in oncology are currently in shortage. Cytarabine, paclitaxel, azacitidine. If you work in oncology, we've definitely heard these names. So as a summary, we have complex trial designs that have a huge impact on supply. We have more uncertain events. We have a complex planning, complex IRT configuration, very frequent monitoring on a lot of factors, and a supply chain strategy that needs to be more resilient to actually match all of these changes. The problem is that complexity generally results in increased waste, increased shortages, or at least for all of us in the room, increased time spent on managing those complex factors and maybe even stress, which we definitely want to avoid. So before we talk about solutions to that complexity, I want to invite Marine to talk a bit about the purpose. Why do we do this? And why will it be so important that we manage complexity in oncology in the future?
Thank you, Amaury. Indeed, I think we all know why oncology is important, because at the end of the day, it is also part of our responsibilities to save patients' life. And this is why we need to keep developing new drugs and being innovative in the way we manage complexity. And as Amaury just explained, the supply chain is already very complex, and we can only expect this level of complexity to increase even more in the future. And the idea behind this chapter is not to give you a 101 introduction on oncology, that's not the goal, but really to take a step back on the why we are doing this and why we are all together on the same boat. Oncology is a difficult disease to understand because it exists so many different types of tumors and cancers that it makes it very difficult to study and to find the right treatment. And again, as Amaury just mentioned, there are many parameters or inherent characteristics to the disease itself that needs to be taken into consideration and that are simply out of control from the supply chain. And because of this, unfortunately, cancer is still the second leading cause of death in the US, but we can also see the same trend in other regions in the world. However, on a more positive note, what we can also observe is the fact that oncology is the world's largest therapeutic area. There is a tremendous number of ongoing clinical trials that are focused on cancer, and it represents more than 30% of the global clinical pipeline for pharmaceutical companies, meaning that we are moving in the right direction and putting all of our efforts where it should be. To make it even more complex, most of genetic mutations that lead to cancer are simply because of random errors and not because of inherited genes from family or harmful behaviors. And in this case, prevention is key, especially when people become older. It is very important to encourage them to do regular checkups and to bring more awareness about healthy lifestyle. And by doing so, we can already decrease the risk by 40%, which is amazing. But of course, this is also associated with an important economic cost for society. Regarding the types of treatments that already exist, this is quite impressive, and we have made some significant breakthrough over the past few years. For instance, we started to use treatment with vaccines or immunotherapy to leverage the power of the immune system to fight against cancer cells. But as I said, even though we've made some significant progress, there's still much work to do, especially when we know that the number of new cases will go over 22 million by 2030, in comparison to 14 million in 2012. So this is a 50% growth over 15 years. And if you look at the graph, you can quickly realize that the oncology market is actually dominated by only few products. We call them the blockbusters. And you can also realize that all the big players of the industry are present and heavily investing in oncology. And this can deliver two different types of message. So the first one being that even if we have some very promising prospects in the clinical pipeline, actually only few of them will turn into potential benefits. And it also demonstrates that there is a fierce competition on the oncology market between the different players of the industry. And speaking about competition, I think it's quite logic, but we can clearly see that the number of clinical trials are on the rise. This is the trend that we can see over the past few years. And to outperform, Pharmaceutical companies are also working on some very specific programs, which is also slowing down the recruitment as it becomes more difficult to actually find the potential candidates. And again, as the complexity increases on these kind of aspects as well that we need to take into consideration, the R&D timelines become also much longer. And if you compare the average timelines for oncology, you will have around 12 years in comparison to only 8 years for non-oncology trials. So knowing all of these and still keeping in mind the why we are doing it, I believe this is a nice segue to maybe start speaking about the potential solutions to make sure that first we can deal with this level of complexity, but at the end of the day, we can also achieve our goal. And for that, I will let Amaury introduce the next section.
Absolutely. So actually, as Marine highlighted, managing complexity in clinical trials, especially in oncology, is becoming actually a competitive advantage. Because if we are able to control that complexity, it means that we have less risks of shortages, less risks of too much spending in a specific clinical trial, and less risks also of having delays in clinical trial timelines. Because, I mean, let's be honest, sometimes supply chain can be a bottleneck to timelines if the drug is not available. And it can happen with low yields, with delays, with anything, any problem that we know happens often in the supply chain. So managing complexity is a competitive advantage. So let's try to discuss some solutions to control that complexity in clinical trials. Well, quick reminder on the complexity factors we talked about earlier. But what we want to share with you today is specifically 6 tips and tricks that we have worked on for years at Insight and used on every oncology trial that we work on, and that we can see work and really, really help reduce the amount of time that we spend managing these trials, the amount of stress from the trial managers, and of course, waste or general complexity in the monitoring and the management. The first tip, and we will illustrate all of these tips, of course, with a case study, each one individually, just to highlight how important they are. So the first tip is really to collaborate with clinical operations on trial design. Because trial design, as we said, impacts supply chain massively. We need to ensure that the design of the clinical trial is also friendly towards the supply chain, and sometimes very small changes can be very impactful. We also need to become more accurate with assumptions. It doesn't specifically mean getting better assumptions. It also means using the assumptions in a better way, as we will highlight in the case study. We strongly recommend using simulations to have more accuracy in the demand planning and the supply planning. And as we're talking about technology, IRT systems, having the right IRT with the right features and configured in the right way, because all of that together would allow having a better monitoring as well through automated monitoring, actually, that report outliers. And to close it all, we will talk about how to make supply chain more resilient to all of the changes that we mentioned before. So Marine, if you want to get started with the collaboration, I think you will be presenting the first case study.
Yes, perfect. Thank you, Amaury. So indeed, the first case study that I would like to show you today is about collaboration on the trial design. But first, I would like to introduce you to the trial a little bit. So in this trial, we have 230 patients. We have 2 different treatment arms. So one with the active and the other one with the standard of care and the vandetanib, which is an expensive inhibitor drug. And depending on where you source it, the cost can be about $9,000 US. So for the vandetanib, there are 2 different kit types. So we have the 300 milligrams and the 100 milligrams. The network is quite complex and the shelf life is also quite short. So we only have 12 months. And as you can imagine, the real challenge in this trial was to actually manage the potential down titration for the patients on the vandetanib. So all patients will start on the 300 milligrams. And let's take the example of a patient that will actually down titrate on 2 milligrams. Knowing that there are 4 cycles visit interval, it means that this patient will receive 2 times 100 milligrams times 4 to cover the 4 cycles. So in total, it means that if we want to cover the potential down titration of this patient, we have to keep at all sites anytime 8 kits as buffers, which is quite important. And another assumption to consider is the fact that actually we expect that only 3% of the patients will potentially down titrate on the vandetanib, which is a quite low number. As you can imagine, the impact on the average and on the cost was quite high. And the solution that we discussed and that we successfully implemented together with the clinical team was actually to authorize an unscheduled visit 2 weeks after the down titration. So the patients would only need 4 kits instead of 8, because he would come back after only 2 weeks and not 4 weeks as in the initial assumption. And this, of course, will allow us to reduce the number of buffers that will need to be kept at site. And by changing this, the impact on the average was about 25% reduction, and the supply cost, we managed to decrease it by 11 million. So we know that challenging the assumptions in the protocol is not an easy exercise, and this is why it is very important to closely collaborate with the clinical operations to really make it possible. Here we could also potentially challenge the patient friendliness, but as I was mentioning, only 3% of the patients could actually down titrate, so meaning that the number of patients that will be impacted is quite low. And more importantly, we had a shortage in the vandetanib, meaning that we had to find a solution to reduce the waste. Otherwise, it wouldn't have been possible to initiate the trial at the first place. And speaking about assumptions, I believe Amaury will also present a very interesting case study.
Absolutely, because collaborating with clinical operations and clinical supply chain on oncology trial means a lot of things. It can mean, of course, trying to have a protocol that matches a bit better the supply chain to try to make it less complex to manage. It can be also talking about the country list or a lot of different things, but it also means agreeing on assumptions. Assumptions such as, in our example, the average treatment length for patients, because we know that in oncology, patients can basically stay for an indefinite amount of cycles until they have disease progression or are actually cured or basically that the treatment is working. So in this specific case study, we had an assumption that was made that patients would survive on average 15 months in the trial. But we know that in the reality, some patients will only stay 1 or 2 cycles. Some patients will stay maybe 30, 40 cycles. It's really hard to know from the beginning what will be the actual treatment length for a specific patient. And in my experience working in oncology, I've seen a lot of forecasts being done with the assumption that, well, as the average is 15, let's just consider that all patients have 15 months of treatment. That is one way to do forecasting and to use the assumption. The other way is just actually accurately modeling patients being dropping out of the trial at each cycle, at each month, with the average being such that the average treatment is 15 months. It's 2 different ways of modeling the same assumption. So even though the assumption is correct, the 2 different ways of modeling will create a different forecast. This is the demand forecast, a cumulative demand for the assumption that all patients do 15 months of treatment. And this is the demand forecast with the accurate dropout modeling and patients dropping out over time from cycle 1 to cycle 50, for example. And we can see really big differences between the 2. If we're looking at specifically the first assumption, which is all patients will do 15 months, what's happening is that we're overestimating short-term demand in that case, because we're saying that no patient will actually drop out in the first 15 cycles, when in the reality, it's true, patients will drop out in the first 15 cycles. So it means that we would be overestimating demand in the beginning and overproducing drug, as well as underestimating long-term demand and long-term survival of the patients. It seems quite basic, but we've seen a lot of forecasts done in Excel, for example, that just simplifies so much the assumption that we end up having an inaccurate demand plan that makes the management of the supply chain even more complex due to higher waste and higher risks in the long term. Another assumption that is typically important in oncology due to weight-based dispensing is the average weight of the patients. So here, I think you all know bevacizumab, the Avastin, typically dispensed to patients on a 10 milligram per kilo doses. In this trial specifically, the assumption was the average weight of patients is 70 kilos. Again, typical easy way of doing it is saying all patients are 70 kilos, which means that we can do a forecast based on that number because it's the average. The more accurate way of doing it is have ranges of weight, basically a distribution of weight from, for example, 40 kilos to 150 kilos, and have this distribution of weight such as that the average is still 70, but that we have a realistic view on the weights of the patients. And it's specifically important in the case of Avastin due to the fact that we have a combination of 100 milligrams of Avastin and 400 milligrams, which means that patients will consume both. So for example, if they need 1,000 milligrams, they will need 2 kits of 400 and 2 kits of 100 to get to 1,000. So this is a forecasted demand with the assumption that all patients are 70 kilos. We have about 8,000 kits of 100 milligrams forecasted and about 7,000 kits of 400. But if we use a simulation solution and accurately simulate the ranges and the distribution, we're realizing that their initial assumption was actually inaccurate. We were overestimating the amount of 100 milligrams and completely underestimating the amount of 400 milligrams that were needed. Because what typically happens in oncology is that patients weighing below 70 kilos will generally have the lowest possible weight is generally going to be around 40 kilos. So there's only a 30 kilo range below 70. If you look above, the range above, it can go from 70 to maybe 150 kilos. The range is much higher. So the demand of 400 was completely underestimated, which can create a lot of risk as well as additional waste and costs in the trial. So again, the assumption is accurate. 70 kilos is very likely to be the average weight, but it's the way that we're using that assumption that makes it so impactful on the forecast and on the management of the trial. And so it kind of links to the next case study, using simulation solutions for clinical supply planning. What I showed before, the way to accurately model assumptions means having accurate simulation of all of these assumptions. It also means that for clinical supply planning, it can really support the accuracy of the planning and therefore reduce risks and complexity. If we're looking at traditional forecasting without simulations, how do we typically do very traditional forecasting? Well, we need to assume the patient demand. We need to calculate what will be the supply chain needs, so buffers on sites and depots, expiry replacements, temperature excursions, and kind of understand what will be the needs in the supply chain that we will, well, not dispense to patients, but actually have there as a safety stock. And then have on top of that a certain overage to cover uncertainty in the demand. That's just the traditional way of doing it. We worked at some point with a client that provided us this basic calculation of 90,000 kits being dispensed to patients. Due to a short shelf life, they expected about 62,000 kits to be lost at sites and depots due to expiry replacements and buffers. And they calculated on top of that a 25% overage just to be safe and manage uncertainty, which brought us to about 175,000 kits. And if we consider that 90,000 is the demand, that was approximately 48% waste. The problem is that in traditional forecasting, as we showed before, I mean, don't try to read the content. We will share the slides afterwards. But as a summary, the patient demand is generally not accurately forecasted in a typical forecasting solution. It's generally overestimated. And there's a lot of aspects like titrations, dropout, weights, strata that are either not possible to model or not possible to model accurately in a typical forecasting solution. So the demand we know are not accurate. On the supply chain side, typical forecasting solutions don't have the power to understand what proportion of the buffers will actually be consumed before they expire, which means that a lot of the numbers that we're considering as site and depot buffers and expiry replacement is very likely we're actually going to be able to consume them instead of just wasting them at 100%. And on the other hand, overages are typically a manually calculated number with a one-size-fits-all. We're going to put 20% on all of our trials just to be safe. But the problem is, if it's too small, it's risky. If it's too high, it's too much waste. And it should be a different value per kit type, per release, per country. The overage should be extremely dynamic as a value. When we run that same trial into a simulation solution, the numbers we got were completely different. First of all, the demand was 75,000, not 90. We got more accuracy in the demand simulation. And then the sum of the overage and the supply chain needs went down to 52,000. We just realized that with the assumptions that we were taking, the kits at sites and depots were very likely to be consumed before they actually expired, which brought us down to 127,000 kits and basically reduced the total amount of kits produced by about 30%. And this is not even specifically due to the optimization functionalities of the system, but just to the accuracy that it brings into the demand forecast. And as we're talking about technology, let's stay on the IRT system. Marine, do you want to take that one?
Yes, sure. Thank you, Amaury. So we know IRT configuration plays an important role in the optimization of the waste and supply budget. And what is difficult when we speak about IRT, and I believe everyone will be able to relate on this, is the fact that each IRT has different capabilities, and some of them are more advanced than others. And especially when we are speaking about oncology trials where the supply chain is so complex, it is really key to choose the right vendor, depending first on your trial specificities, but also the values that the IRT will be able to suggest. And the collaboration with an IRT is really to be able at the end to implement the best possible IRT strategy for your trial. Now I would like to illustrate this with two different examples. The first one is related to the values of the short window and the long window. In most IRT systems, you will be able to encode only one value for all countries in your network, meaning that you will have to consider the worst case scenario for each country, which is driving your overage and costs negatively. But by changing this configuration and allowing different values depending on the shipping lead times that you will have to the sites or the visit intervals that you will have depending on the treatment of your patients, you can adapt the short window and the long window to exactly what you need to keep as a safety stock. And you can also optimize the number of expiry replacements by keeping the do not count and the do not ship flexible. And by doing so, by having this flexibility in your IRT system, you will be able to have a positive impact on the average. So here in the example that we choose is about 20%, but also you will be able to achieve some significant cost savings. Now I was speaking about more advanced IRT capabilities, and this one is actually a good example. This is a trial where we used pomalidomide, and we had different dose levels. And I think we all know that the pomalidomide is an expensive comparator, and managing the potential down titration and the level of buffers that you need to keep at sites can be quite challenging, especially when you look at the number of sites that we had in this specific trial, and that you look at the cost of sourcing. To reduce that cost, the solution that we found was to actually implement what we call dynamic buffers. And this specific IRT strategy means that depending on the current dose of each patient, the IRT system will automatically resupply the sites with the next dose on which the patient could potentially down titrate. So to give you a more concrete example, at the beginning of the trial, before the first patient in, all the sites were seated with only 400 milligrams because we knew at the beginning that all patients could only receive 4 milligrams. At the randomization, we started to seed the site with 3 milligrams. And only once we had our first patient on the 3 milligrams, we actually started to seed the sites with the 2 milligrams and so on and so on. Of course, the impact on the waste and the cost can only be positive. But I don't know if you remember that when we are speaking about sites, you also have different statuses. So we have the low, the medium, and the high. And the level of complexity of managing those different statuses can be quite difficult. And with the strategy of dynamic buffers, you will not need to do that anymore because it's something that will be automatically managed by the IRT. So here we had a positive impact again in terms of average and in terms of cost, but we also succeeded to reduce the level of complexity to manage the buffers at the sites. And speaking about managing and maybe monitoring, the next case study that I would like to show you is exactly about the same topic. Is how can we automate more our clinical trials by using the right technology? And what we suggest is to actually perform risk-based monitoring thanks to the simulations. And indeed, with the simulations, you are actually able to capture all the variability and all the uncertainty that you will have in your clinical trial. Meaning that you will not only have your average patient demand forecast, but you will also have a minimum and a maximum that you can see on this graph with the orange bar. And every time you will have an outlier, you will be able to monitor it in comparison to this minimum and maximum to understand first, do I have a risk or not? And then most importantly, by when this risk is coming. So you can anticipate it and take actions proactively. And if I take a more concrete example, this is a trial on which we closely monitored the recruitment. And these were actually the initial assumptions that we received. And then we received updated assumptions, and the recruitment was actually postponed by a couple of months. And then we realized that even though the recruitment was postponed, it went a little bit faster than expected. And finally, we finished the recruitment in only 9 months rather than 20 months. Again, I would like to emphasize here that if we didn't have this risk-based monitoring, we would probably have been too late to identify this risk, and we couldn't have avoided potential risk of stockout. And so here it's a case study where we focus exclusively on the recruitment. But of course, this is not the only parameter that you can monitor, especially in oncology trials where you have so many different aspects that can come and influence your supply. You can also monitor the dropout. You can also monitor the titrations or the weight, everything possible to make sure that, again, you can manage and monitor your trial without having any risk. And now I would like to let Amaury maybe explain the last case study.
Thank you very much, Marine. Indeed. So monitoring is very important because it allows us to understand outliers before they actually cause problems inside of the clinical supply chain. But then when we have monitored and we identify the outliers and we need to react and change our strategy to adapt to the real-time events, which is true in any clinical trial, but is going to be more frequent in oncology, it also means that our supply chain needs to be a bit more resilient in order to react to those events. So let's just illustrate that with one final and last case study. We had a trial here with a comparator that had a 9-month shelf life after QP release, so quite short. And due to a limitation with their CMO, this client wanted to do, was only able to have releases twice a year. And they were considering maybe changing vendors to have a bit more flexibility on this. What happens is, let's actually take Marine's example from earlier with the demand simulation. And this is basically what happens in any clinical trial as well. If you have your first release in January and your second release in July, that's one release every 6 months. So that's basically the case study that we just showed. Lot number 1 will expire in September, 9 months later. And what does it mean in terms of the quantities we need to produce? In lot 1, we will need to produce quantities to match the worst case demand by the time that release number 2 is going to happen, which means about 700 kits, which, as you can see, is an intersection of the worst case demand forecast in July 2020. So we need to produce about that amount of kits, 700. But it also means that if we're not on the worst case demand, we only have this period of time from July to September to consume the overstock of lot number 1 that was not consumed. So if we are actually on the bottom demand line, the minimum demand line, we will probably waste a lot of drug because we only have 2 months to consume the overstock. It also means that we have less chances to react to uncertain events because there's less chances to postpone a lot release or to increase the quantities of the next release because that release is going to come a lot more into the future. So it's also more potentially risky for the trial. If we now look at the same example with a different packaging schedule or sourcing schedule, if we did source every 4 months instead and have lot number 1 in January, lot number 2 in May, we now can release less kits on lot number 1 because we need to only cover the worst case until May 2020 instead of July, so 400 kits. But it also means we have a longer period of time after the release of lot number 2 to consume any overstock from lot number 1. It is therefore in this scenario very unlikely we will have any waste in lot number 1 because we will have had time to consume it before it expires. Even if we are in a much lower demand or if the recruitment was postponed, it is a lot more efficient and more flexible. Let's just illustrate with numbers in that specific case study, and it's quite scary numbers. If we were to release every 6 months due to the presence of countries such as Argentina and Brazil, it means that we would have had a $262 million sourcing cost because a lot of the drug would go to the garbage and not be used. Meanwhile, if we produce every 4 months, we can cut that in half. And every 2 months, we could cut that in 3, basically a third of the initial cost. So this is not really a question of saving money again. It's a question of reactivity. The more frequently we package in oncology, the more easily we can react to all of these uncertain events. So yes, it saves money, but mostly it allows us to better control risks in the clinical trial. Now, let's try to summarize a bit everything we talked about. Marine, do you want to take that one?
Yes. Indeed. So to conclude this webinar, we would like to give you some key takeaways that we hope you will be able to bring back to your organization. But first, I would like to come back on the tips and tricks that we used to drive our case studies. Each of these case studies actually represent a success story that we had with some of our pharma partners, and that can be implemented in every organization. However, it requires to put in place some good practices as always, and this is what we would like to show you as last slide. So we identified 4 main good practices. And the first one being implementing and defining the right processes. So I think we all understood oncology trials are very complex, and we need to have specific processes that will allow us to put in place the right collaboration between the clinical operations and supply teams, but also a process that will allow us to collect and to challenge the assumptions that we have and to make sure that we can continue building on the initial information that we received. As you know, there are a lot of different technologies out there, and it's not always easy to find the right one for your organization. But it is truly important to choose the technology that will provide you with accurate and robust forecasts on which you will actually be able to base your decisions. IRT configuration, again, very important to include them in the discussion to make sure that we can implement the best IRT strategy that will fit your trial needs. And integration capabilities are also very important, because as you understand, it is quite important to be able to capture information from the past data and from the actuals to make sure that we can narrow down what will happen in the future. And last but not least, monitoring your trials, again, with simulations by taking your decisions not only based on the average, but on this cone of confidence that you will have between your minimum and your maximum, to be able to anticipate any deviations and to be able to react proactively. So if today you have to take home some key messages, we hope that those ones will resonate to you. If you have any questions, don't hesitate to ask them in the chat. Amaury and myself will always be available also after the webinar if you would like to reach out. Again, don't hesitate. And thank you so much for being here today.
Absolutely. And thank you very much, Marine, for the summary. I know a lot of the numbers that we showed here can seem quite shocking. I mean, it's big numbers. So I also want to invite everyone to reach out in case you're either skeptical, of course, or interested in this. We would be very happy to prove everything we just showed by doing a pilot or whatever, to just let us know. But now we actually have 12 minutes to answer questions, and I see the chat is actually flowing with questions, which is amazing. So let's start with the first ones that were asked. I think there's one here. Okay, so there's one about the case study on the collaboration with clinical operations and clinical supply. When we suggest a visit after 2 weeks, was it under the assumption that all direct-to-site shipments lead times were less than 2 weeks? If we had a country with a longer lead time, would we have a different strategy over there? Absolutely. So in that case study, so patients were actually coming back to the site every 4 cycles. So they were coming back every 3 months approximately, which meant they needed to receive 8 kits of the down titration dose in case they were to change doses at some point. What we did there is to say, let's only give them the amount of 1 cycle and keep on every buffer of every site 1 cycle of buffers, and have those patients come back in an unscheduled visit to get the rest of the kits, which really reduced the amount of drug that we needed to make the trial feasible. And indeed, if we had a country, which was not the case here, with a longer lead time, we'd probably increase a little bit the buffer in that specific country and have the unscheduled visit happen a bit later. Another question here about the last case study we showed. How can we supply the rest of the year if we can only produce 2 campaigns per year, especially if we're bringing the second campaign earlier in the year? So it's a good question indeed. And it was really the decision of the client to actually look for another CMO that had more flexible capacities on their packaging chain. So if they were to package every 4 months or every 2 months, they would have to change CMOs to a CMO that actually has that capacity indeed. So remove that constraint of producing only 2 times a year. It was a very important decision to take. What would be the criteria to look out for in an IRT that would be considered as a strong IRT? Marine, do you want to maybe take that one?
Yes, of course. So I think it's very important to emphasize that there is no right or wrong IRT. Every IRT system have their own capabilities, but what is really important is to be able to test the impact of choosing that IRT depending on the specificities you will have in your clinical trial. And for that, optimization tools can really help you to get the data that will allow you to understand what if this IRT doesn't have that specific capability? What will be the impact in terms of waste? What will be the impact in terms of supply budgets? And those kind of assessments are very, very important, especially when you're before you are selecting the IRT vendor, because even if it sometimes requires some customization, sometimes the positive impact again that it will have on the waste and supply budget will be worth the cost of the integration.
Indeed. And so, I mean, just to give a couple of examples of criterias, we illustrated a couple in the case study. The do not ship, do not count, do not dispense values must be flexible per visit per country, and not be static values. It's one example. We talked about the prediction values that should be flexible again per country and depending also on the visit window. It's a very basic example, but if the short window cannot be adapted based on the visit window of the trial, and the visit window is plus minus 30 days at some point, it means that your short window will need to be extremely long, and it will increase your waste and the amount of inventories for your investigators at the site. These are two examples out of multiple different features in the IRT that are really important to look at and that will influence and impact supply chain massively, and that unfortunately very little people know the impact of. So if you want to further that discussion and maybe have a more exhaustive list of examples of features to look for in IRT, please feel free to reach out to us afterwards. We have more time for more questions. So what is the typical overage for an oncology trial? I think that came from the example on the typical forecasting solutions. Well, it's a question we get very frequently. What is a good overage to use? The answer is not that easy, is that every clinical trial design will need a different amount of overage. Some trials need 5% of overage or 2% of overage. We've had a couple of those. And some trials need 300% of overage. And it's very hard upfront to tell you what is the right number for each trial. It's important to have a system that can assess extremely accurately how the trial design impacts the overage. If the overage is manually calculated or done with a rule of thumbs, we know it's not going to be the right number, and underestimating that number can be very dangerous, just as overestimating means that we're going to, well, throw money through the windows, basically. So it's a very hard balance to strike. Okay, one question. How do you convince ClinOps to let supply influence the protocol? That's a funny one. I mean, Marine, do you want to maybe try that one?
So protocol design and trial design has always been a little bit challenging, right? And again, I believe it is really important to leverage data to make sure that you can have visibility on the impact of some decisions. And that will be key if you would like to improve the collaboration between the cleanups and the supply team. And showing the impact on some decisions and how you can ensure the trial feasibility by challenging the protocol design together. Using data for this, I believe it's really key to just make sure that that you can collaborate by just looking at the same data. So I don't know if Amaury, you have something to add on this, maybe.
Yeah, it's an interesting process because indeed it is not frequent for pharma organizations to have clinical supply influence protocol decisions, but it is something that generally moves step by step. It starts with one case study by having data-driven decisions, so we understand what is the impact of the changes, both on the patient, on the trial, patient friendliness, and on the supply chain, have numbers like we showed in the case studies, and start a discussion with the clinical operations. Sometimes, like in our case study, just making those modifications is exactly what we need to just make the trial feasible, which is also the objective of clinical operations. So we need to find common ground, common objectives, and have data to illustrate the impact of all of these changes.
And maybe to just add one last point on that specific topic, again related to the good practices that we just mentioned, having a process a dedicated process for trial design where you can make sure that both the cleanups and the supply team will be involved is also a good starting point to make sure that everyone will be involved at the right timing.
I think we might take a last question here. So someone has a shortage on paclitaxel in their trials. How can I ensure that my trial is not impacted too much by the shortage? It's a very good question. You might have heard indeed that one of the manufacturers of paclitaxel had some quality issues and had to actually stop production and close their sites for a while, which means that the whole industry is basically focusing on only one manufacturer who doesn't have all the capacity right now. The solving a shortage is a whole lot of work. I mean, we have a whole webinar that lasts for 1 hour explaining how to solve a shortage. I would really invite you more to contact us and maybe have a separate meeting on that very specific topic. There are hundreds of ways to solve a shortage. It generally involves prioritizing solutions, quantifying the impact of the solutions on the risk, but it also mainly means that we need to have a solution that is able to accurately understand how much risk we have, how many patients are impacted, what kit types are impacted, where the risk is happening with extreme accuracy, which our solution does, of course. Unfortunately, we have had to save some trials from shortages in the past. We have a whole method to go through that. So please, let's maybe have a follow-up meeting on that topic. And so before we close the meeting in 2 minutes, we might have some time for additional questions. Any other questions here from the audience? Okay, so it was really great to have you all join us today for this webinar. Thanks so much for your participation. And we are very happy to follow up with you after the meeting if you have any follow-up questions or if you want to try out everything we showed. The slides will be shared on demand, so let us know if you want to receive them. And the recording of the meeting will absolutely be available as well. Thanks again, everyone. Wish you a very nice day or a very nice afternoon, and see you soon.
Thank you, everyone. Have a good day.